So exactly. I mean, it seems like that perhaps combinations are in the future rather than single agent. Is that your take on it, Krishna?
Yeah, I think that, we talked about this potential synergy between the post-transplant milieu and checkpoint inhibition therapies, and similarly, other novel agents like targeted inhibitors might also synergize with checkpoint inhibitors. There's actually a lot of excitement at this meeting about preclinical data that was presented in the colorectal cancer setting, where, and I think Roche is planning a trial of a combination of cobimetinib, which is a MEK inhibitor, with the RAS pathway being active in a lot of colorectal cancers, and also active in a subset of AMLs and CLL, and in combination with atezolizumab, which is a recently approved, again, checkpoint inhibitor, but against PD-L1, one of the ligands, as opposed to PD-1. Now, the only other thing that I would add about that is we've had some interesting studies done in my laboratory leading to a couple of recent papers where we demonstrated that the RAS pathway is also important in IL-2 production in T cells, and we actually showed that MEK inhibition actually inhibits naive and early memory T cells, but spares the antigen-specific cells that I would predict would be important for malignancy control, and we know that are important for the control of chronic viruses. So we have to be careful. Some of these targeted inhibitors, while they may actually lead to the death of cancer cells and lead to the exposure of antigens that may be good, in terms of inducing an immune response, that they may actually affect the effector T cells themselves. So I think obviously we should have good preclinical models leading to clinical trials, and then we have to look at obviously both safety and efficacy, and that needs to happen.
I think the combinations, we see combinations and data at ASCO update in melanoma, where it's very, very impressive EP niveau in melanoma. I mean, the results are really outstanding comparing to single lesion. And to the point that when we started talking about the mechanism of the checkpoint inhibition to regulate immune response is a very complex mechanism with multiple checkpoint gate control, gatekeepers. And so there's at least 10 different antibodies potentially, receptors that we could target. So it makes sense that inhibiting one antibody, one receptor is not enough. So the data, by extension from the data in melanoma that is also now true in lung cancer, I think this would make a lot of sense for us to move in the heme malignancies as well in combination early on.
Yeah, how about the T cell redirecting therapies like the bispecifics? I mean, certainly there are trials on the books looking at trying to combine those molecules with checkpoint inhibitors. Any thoughts there?
No, I think that makes sense as well, because again, similarly, you have a cellular mechanism of action, and then I think if you have again the T cells that may get engaged may be in a maturation state or may be inhibited, and so you may need not only to get the T cell there with the bispecific agent that, for example, like blinatumomab brings CD3 to the point of the tumor, but you may actually need to change the functional state of the tumor with the checkpoint inhibitor. I think in that context, it's just worth mentioning that there are some of the other checkpoint inhibitor therapies that we haven't yet seen translated. LAG3 or CD223 is also another inhibitory receptor on the surface of T cells. It's also present on regulatory T cells. It turns out that the adenosine pathway can actually have a negative effect on T cells, and there's a company, Corvus, that's developing therapeutics against this novel approach of doing checkpoint inhibition. And so I think we're going to see not only, I think, all of these interesting things like where is the timing, how does this synergize with either transplant or combination therapies, but we'll also see novel agents that may have different patterns of activation of the immune response. So it'll be an exciting time.
I think you mentioned the blinatumomab or the BiTE antibodies. There's many others. The BiTE antibody for our audience is actually a small molecule that is a combination of a recognition of a CD3 and CD19, for example, for blinatumomab. So as you mentioned, Krishna, bringing them together and forcing the engagement of the T cells. And that's a way to release the T cells and engage them. And the responses have been amazing in lymphomas, including in refractory large cell lymphoma. And it has some issues with, because it's a continuous infusion program, so it'll be tedious, but amazing responses. And it has a similar toxicity as we see in CAR T cells with the CNS toxicity, because T cell activation goes to the brain. But at the same time, we see responses in ALL. In ALL, we actually get patients that go into MRD negativity, and that's where it is approved in ALL now, in refractory ALL. So this is really something that is going to be in situation where you don't have a CAR T cell possibility. That could be a great backbone to try to build up, and maybe on a BiTE antibody plus a checkpoint inhibitor. I think that would be very promising.